Hello everyone, my name is Jeroen Ingalls and I'm a research faculty at the Florida State University Coastal and Marine Lab. And today I'd like to present to you our research on why loggerhead sea turtles are meiofauna abundance and biodiversity hotspots. Now I have to say that this work couldn't have been conducted without the generous support of various funding organizations and the help of a number of collaborators and students and volunteers, especially with help in conducting the field work. Now, when people look up images on the internet of sea turtles, this is often what you're presented with, a nice, clean, happy sea turtle swimming in the ocean. But as a field biologist, it is much more likely to be encountering a sea turtle that looks like this, whereby the carapace or the shell of the turtle is covered in many different types of organisms. And when you have a closer look at these, it becomes apparent that, one, the abundance of these organisms can be very high, and also that the diversity of these organisms can be very high. Our two main objectives for this research study was, one, what is the structure, diversity, and functioning of loggerhead sea turtle epibiotic meiofauna, and also to compare with some of the results we've published in previous studies on hawksbill turtles, and to compare to see whether loggerhead sea turtles have different communities and whether the abundance and diversity is higher or lower. And then secondly, we wanted to find out more about whether meiofauna epibionts can give us more insights into loggerhead ecology and vice versa, whether loggerhead ecology can give us more information on meiofauna epibiont ecology. Now, the two main topics of this objective is, one, tackling the meiofauna paradox. Now, meiofauna live in between the sediments normally, and they don't have pelagic larvae, so they don't have an active means of dispersal. They live their entire life cycles in sediments normally. Yet at the same time, we find that many species and many groups of meiofaunal organisms have very wide distributions. So it seems contradictory. No active means of dispersal, yet wide distributions. So by looking at the epibionts of turtles, we wanted to get an idea of whether there's a potential for phoresis or meiofauna traveling on the backs of turtles for large distances, and whether we can get more insights into the question of the meiofauna paradox that way. And then secondly, we wanted to dig into more of the question whether we can use meiofauna to understand more of the sea turtle ecology. Our study site is situated in Florida, shown here on the map. More specifically, a barrier island called St. George Island, which you can see here. It's about 28 miles long, and at its widest point, it's about a mile wide, and it's connected to the mainland with a very long bridge. It's basically a strip of land with a very long beach, and it is part of the Northern Gulf of Mexico recovery unit for loggerheads. There's a high density of loggerheads nesting here every year. So that was our study site. And during about 2 weeks in the peak of loggerheads nesting season, we have a team of researchers that go out and every night from about 8 or 9 in the evening until 4, 5, 6 o'clock in the morning, we walk this stretch of beach numerous times with different teams on the lookout for loggerheads that nest on these beaches. Now, we have to say that nobody should try this at home. Turtles are wild animals. They like to bite and slap when encountering them. And you need training and permits to interact with them. And our work was all conducted under the various permits that allowed us to do this research. Now, these images were taken during the summer, so we're not allowed to interact with the turtle or start our sampling until after the turtle has completed her nest. Now, also, I have to mention that these pictures were taken under red light, even though they're black and white. We don't use white or yellow light, so not to disorientate the turtles in the process. And we use putty knives to take all the epibionts off and then collect them in bottles with preservative. And this is what a turtle looks like when it's finished the nest and it's on her way back to the ocean. You can see some coverage with algae, but lots of sand also. The turtle digs in her nest, and there's lots of sand that ends up on the carapace of the turtle. And you can see some barnacles here. And this is a turtle on the right here that this is what the turtle looks like when we've finished the sampling process. So it's an exhaustive process where we literally take everything that we can find carefully off the turtle shell to get a complete view, a comprehensive view of the communities living on the back of these turtles. Now, this image on the left here is a study that was published a few years back, which suggests that 9 different sections that one could sample for epibionts. Now, we quickly realized that wasn't really possible, so we separated the turtle into 3 sections, the turtle carapace rather, into 3 sections: anterior, a middle section, and a posterior section. And we sampled a total of 23 turtles. And this is a very complex table, lots of numbers, but it's basically just to show you that on the left, you can see all the different turtles that we've sampled. And in the bottom right corner here is the total number of organisms, meiofauna organisms that we found on these turtles, over 800,000. And you can see that the total numbers are pretty astonishing, really. A huge amount of organisms were recovered from these turtles. It really shows that they are hotspots of abundance. There's a huge amount of organisms living on the backs of these turtles. Now, these box and whisker plots show us the total meiofauna abundance there, the total nematode abundance, the higher taxa meiofauna richness, and then the nematode genus richness down below here. And we see that they're between the different sections, anterior, middle, and posterior, there's not really a difference in terms of abundance between those sections, and neither is there a difference on higher taxon level for meiofauna in terms of richness of those taxa. But when we look at the nematode genera diversity, we can see an increase towards the posterior section. The back of the turtle often harbors greater diversity of nematodes compared to the anterior sections. Now, when we look at an NMDS plot of meiofauna community structure, we run a statistical analysis to see whether there are different groups of epibiont communities, each linked to their own turtle. You can see how there are 2 distinct groups. The group on the left here has very few Nopli larvae, whilst the group on the right here has a much higher abundance. So there's 2 distinct groups based on the meiofauna epibiont community structure. And this is represented in this bar chart as well. These turtles here, represented by their ID number, have a huge amount of Nopli on the back of their shells, compared to a rather more diverse community structure in this group of turtles here. So 2 distinct groups when we look at the higher taxon meiofauna level. Now, when we look at nematodes, and in these plots, we look at nematode diversity. So each plot represents a different type of diversity index with various levels of importance given to abundance or pure richness. So for all of them, we could see that the diversity and evenness increase towards the posterior. So a clear indication that there's more diverse nematode life on the back of the posterior side of. Now, when we run an NMDS on nematode genera community structure, and we look for significant differences between groups of samples, we see that our analysis splits up 4 different, distinctly different groups of epibiont communities. And what we are trying to figure out now is whether that is actually linked to behavior or potentially population differences of the turtles themselves. We have a PhD student, Ian Silver-Gorgeous, who is currently working up isotope data and genetic data of the turtles, and see whether there are links between the results that we have here, the different epibiont communities, and whether there are links between those different groups and the actual turtles themselves. Now, if that is the case, then that would suggest that there is a strong link between what lives on the back of a turtle and the ecology or population difference between the turtles themselves. So in summary, we found huge abundance and diversity on loggerheads, much greater than we found on hawksbills a few years back, and that can be explained by the difference in behavior of the turtle. Loggerheads are informal miners. They dig up sediments to feed, whilst hawksbill turtles forage more around coral reefs and are more pelagic feeders. So distinct difference there. It creates this much greater opportunity for meiofauna organisms that are swept up with the sediments to land on the back of a turtle and establish a community there. We also found distinct meiofauna and nematode community structure and diversity differences between the different sections of the turtle. That could be explained by various reasons, one potentially being flipper disturbance or increased water flow in the anterior region versus the posterior region. We found distinct meiofauna communities, splitting up the group of turtles that we've studied in two distinct groups. And when we look at the nematode communities, we see 4 distinct groups. And we are now trying to relate that to the identities of the turtles and see whether distinct epibiont communities are linked to distinct turtle populations. And of course, we found that meiofauna dispersal is likely. The huge numbers that we find on the backs of these turtles, thousands, I think there was one turtle where we found over 140,000 organisms, which is a huge number. Now, if you consider the total number of turtles, the number of organisms that live on these turtles, the large distances that they migrate, and also the fact that their evolutionary age of turtles is over 120 million years. So we have a very long history of potentially meiofauna hitchhiking rides over large distances between coastal areas in large numbers. And this really gives us an insight into how the meiofauna paradox might not be that paradoxical. There is a great potential for exchange between remote locations. And with that, I thank you very much.